Hello, my dear students. Welcome to Science Era.
Have you ever wondered how scientists can quickly tell different types of bacteria apart under a microscope? Today, I'm going to show you one of the most important techniques in microbiology, Gram staining.
What is Gram staining? Gram staining is a laboratory technique used to classify bacteria into two main groups, gram-positive and gram-negative. It was developed by Hans Christian Gram in 1884.
Importance of Gram staining. Now let's talk about the importance of Gram staining. Gram staining is one of the most basic and essential techniques in microbiology laboratories. It is widely used as a first step in identifying bacteria. This method helps scientists and lab professionals to quickly differentiate between gram-positive and gram-negative bacteria, understand the general characteristics of bacterial cells, guide further laboratory testing and identification methods. It also saves time because results can be obtained rapidly under a microscope. Due to its simplicity and effectiveness, Gram staining is used in microbiology labs, research studies, educational laboratories. So even though it is a simple technique, it plays a very important role in bacterial classification and study. Principle of Gram staining. Now let's understand the principle of Gram staining. The principle is based on the difference in the structure of bacterial cell walls. Gram-positive and gram-negative bacteria have different cell wall compositions. Gram-positive bacteria have a thick peptidoglycan layer. Gram-negative bacteria have a thin peptidoglycan layer and an outer membrane. During the staining process, crystal violet enters all cells. Iodine forms a complex with the stain inside the cells. When alcohol is applied, in gram-positive bacteria, the thick cell wall traps the crystal violet iodine complex, so they remain purple. In gram-negative bacteria, the outer membrane is disrupted and the thin layer cannot retain the stain, so it gets washed away. Finally, when safranin is added, gram-negative bacteria take up the counterstain and appear pink. Gram-positive bacteria remain purple. So the entire principle depends on how the cell wall structure reacts to the staining process.
Inoculation loop, Bunsen burner, staining kit, crystal violet, grams iodine, 95% ethanol, decolorizer, and safranin. Distilled water, wash bottle. Microscope, 100x oil immersion. Procedure, step by step. 1. Smear and fix. Prepare a thin smear on the slide. Air dry and heat fix by passing it through a flame 2-3 times. 2. Primary stain. Add crystal violet. Wait for 1 minute. 3. Wash and dry. Wash gently with distilled water. Air dry the slide. 4. Mordant. Add grams iodine. Wait for 1 minute. 5. Wash and dry. Wash with distilled water. Air dry again. 6. Decolorize. Add 95% ethanol drop by drop for 10 to 20 seconds until purple color stops running. 7. Instant wash. Wash with distilled water immediately to stop the reaction. Air dry. 8. Counterstain. Add safranin. Wait for 45 seconds. 9. Final wash. Wash with distilled water. Air dry completely.
Results. Gram-positive, purple-blue. Example, Staphylococcus. Gram-negative, pink-red. Example, E. coli.